Welcome friends, welcome to my channel Plant Science for You. In this video, we will be dealing with the second part of the previous video where we have discussed about the introductory plant breeding, different activities of the plant breeding and history of plant breeding. In this video, we will see different objectives of the plant breeding, different achievements which we have achieved through the plant breeding and different undesirable consequences of plant breeding. We start with this quote by Lewis and Christensen that a successful variety is one with a total balance of traits that makes it more profitable for the growers than any other one they might choose. That means whatever they choose, it will be more provide a more balanced trait and will be more profitable to the farmers. So in the previous video, we have talked different activities of the plant breeding. Like if there is a, we have in the first step we have seen the domestication, where we domesticate our uh, wild species and bring it in our human management. On after that, we practice our different practices like selection, hybridization, etc. After this processes, if there is a less genetic variation present, we go for creating genetic variation using these methods like natural methods like domestication, germplasm collection, introduction, naturally existing variability. These are all present in the nature and we utilize them to create the natural genetic variations. Otherwise, we can use the hybridization process by including intervarietal, distance, somatic, etc. Then we go for mutation breeding too, where we treat our plants with X-ray, gamma ray, UV ray, etc., and go for mutation breeding. Then we go for polyploidy breeding, where autopolyploidy and allopolyploidy plays the most important role. Then the soma clonal variation, and the last one is the genetic engineering. When we what we do now, what we do, we include the DNA or our desired DNA from an unrelated organism to our desired plant crops. After creating the new genetic variability, we go for selection. We select our desired plant, then we evaluate them with the existing variety. If it overcomes the yield outcomes, the yield of the existing variety, then we go for its multiplication and we distribute to the farmer for the for our use purpose. These are all the activities which are performed in plant breeding. If we see, moreover, that what are the main objectives, what are the objectives of the plant breeding, what are the objectives of the plant breeding, we will find that the chief objective is of the plant breeding is to develop such improved varieties of the crop plants that will be commercially successful. That means it will give us more income and as well as it will feed our belly. However, the improvement of in some specific traits, that means we go for the specific trait improvements in subcrops like in rice the milling quality, in wheat the baking quality, etc. Different qualities we will see all of them. These are also the objective of various agronomic, economic, etc. reasons. If we see what are the main objectives, we will find that there is the higher yield is the main objective. Then there may be high improved quality, disease resistance, maturity duration, agronomic characteristics, photoinsensitivity, elimination of toxic substances, stress tolerance, varieties of new seasons, seed dormancy, determinate growth, and synchronous maturity. We will see one by one. The first and the foremost one is the higher yield. It is the main objective. If we see, it is the main objective of any breeding program. If you are planning a breeding program and you are not considering the yield as a your criteria, you are misleading. That means you whatever your focus is for the breeding, you must have considered the higher yield, because you if the crop variety you develop, it is not the higher yielding, then the farmer will not give you importance. That means your prepared variety will have no use. This is the higher yield is achieved by developing more efficient genotypes using breeding program like hybrid varieties. is one of the techniques by which we have developed or increased our higher yield. Like in maize, jowar, and bajra, we have developed such varieties which are very much efficient in yielding this high amount. After the yield, if we have to consider the second one is the improved quality. That means quality. Okay, the quality of plant produce determines its suitability for various uses. That means if we have developed a variety like for like rice, if we have developed a variety and its cooking quality is very poor, then we will not choose. It's high, it is very famous for the high yield, but it is not giving the desired quality. Then you will not prefer. Okay. Likewise, in case of wheat, there is some such quality traits like grain size, color, milling, and baking qualities, etc. In barley, also there is a melting quality. In case of vegetables, there are some definite uh, qualities like size, color, flavor of the fruits. Like in case of tomato, there is a keeping quality also plays an important role. In case of cereals and legumes, there are protein content is also a good measure of the quality. In case of cereals, they are the lysine deficient. So lysine content is also one parameter. Same is the case of the pulses. They are methionine and tryptophan are deficient. So if they are producing more tryptophan and methionine, they will be more desirable. Okay. So this improved quality will be our second objective of plant breeding after improved yield. After that, if we see there is a disease and insect resistance, it is also one of the most important criteria for breeding. Resistant varieties are often the cheapest and the most convenient method to increase the yield as well as the disease or insect management. For example, rust resistance in wheat. You know that the rust in wheat is one of the major player which is diminishing its yield a by a huge amount. And if the only we can use a variety of ways, the mechanical control, biological control, etc. But if we make the variety resistant, we can adopt a lot of things like cultural practices. But if we adopt the cheapest one, which is the resistant variety, that means the genotype of the our variety will be resistant to the rust pathogen, and that will be the most convenient method to yield more and more, and provide an rust resistance in those varieties. These resistance varieties are not only increase the production but also stabilizes its production. Okay. After that, if we see there is a change in the maturity duration, that means it will provoke. Maturity means a crop which we have sown, it will mature in time. It will take about 3 to 4 months depending on the crop. And if we alter the maturity period, that means if we in some cases to fit one extra crop in our crop scheduling, we can use the early varieties. Okay, so for making those varieties, we need to change the maturity duration. It permits the new crop rotation and often extends the crop area. That means it will give us the uh, flexibility to plant three more than three crop in our crop program. And development of wheat variety is suitable for the late planting, which has permitted us to permitted rice wheat rotation. Okay, if we can that rice is grown in the monsoon season, and it will takes about October November, and that is the season for the wheat sowing. So if we can sow our wheat variety something late, we can get more our desirable rice. Okay. Thus, the breeding for the early maturing varieties or varieties suitable for different dates of planting may be important objective in many cases. After that, if we see the what are different agronomic characteristics, that means we can modify different agronomic characteristics such as plant height, tillering, branching pattern, erect and trailing habit, etc., which is often desirable. Such as, for example, in case of cereals, it is desirable that the dwarfiness, because it makes them more fertilizer responsive and nutrient responsive. You know that the earlier wheat varieties are very much tall, and if we give them nutrient, they will become very tall and they will lodge down. This was the main problem. After the Green Revolution, we have got the same semi-dwarf varieties. We have go for the semi-dwarf varieties, which are very much nutrient or fertilizer sensitive, and they can tolerate high dose of fertilizer and grow more yield. These make them lodging resistance as well as fertilizer responsive. So in those cases, the dwarfiness or the plant height is one of the major breeding objective. In case of sugar cane, if we see, there is the long height will be the major desirable characters. After that, the photoinsensitivity. Development of photoinsensitivity. That means it will not depend on the photo period. On thermosensitive, that means it will depend on the temperature. It will, in case of wheat and photo-intensive rice, that is the Oryza sativa variety, has permitted their cultivation into new areas. That means you know that the rice is now cultivated in Punjab, which was not possible earlier because of this photosensitivity and different issues. And wheat is now also cultivating in West Bengal. Previously, it was not cultivated due to this photoinsensitivity. That means they are only grown in case of winter season. But in West Bengal situation, other crops like potato is also there in the Rabi season, and and other issues also there like photoinsensitivity and thermosensitivity. For those reasons, wheat was not earlier grown, and but now this is also solved, and these wheat varieties are grown in West Bengal situation. In case of wheat, the photoperiod insensitivity is due to the two genes. There is a PPD1 and PPD2. That is the uh, photoperiod sensitive uh, photoperiod insensitivity genes, and they show the polymeric gene interactions. If we see the synchronous maturity, that means which is highly desirable in some crops like moong, where we have to go for several pickings is necessary. If we go for synchronous mature the crops and synchronously, then it will have more profitable yield to us. After that, the non-stuttering characteristics. That means it would be very great. It will add a great value to the crop like moong, but the stuttering is a major problems. And if we see that the crop when it is growing and sometime later before our pickings, uh, the fruit is breaking and it is going to the going drop to the going down to the soil, it will know use. So non-stuttering characteristics is one of the major desirable objectives of the plant breeding. After that, the desired determinate growth. That means development of varieties with determinate growth. If we see here that the determinate growth, that means the reproductive structure will end up the crop. And in case of indeterminate, the crop will grow up, and the vegetative growth will suppress the reproductive growth. So in such cases, the determinate growth is desirable, like in case of moong, Fijian pea, and cotton, etc. After that, the dormancy. What are the dormancy? That in some crops, the seed germination before even before harvesting. That means seeds germinate before harvesting, which is our not actually desirable. That means we need to make sure that the seed must have some dormancy, like in case of moong, barley, etc. A period of dormancy in such cases will check the loss due to germination. In some other cases, however, it is not desirable, and there we need to remove the dormancy. After that, if we see the moisture stress and salt tolerance, development of varieties for rain-fed areas. That means where the rain is always happening, so rain is more. There we need some moisture rain-fed areas and for saline soils would be helpful for increasing the crop production in uh, India. That means there where there is a moisture stress or salt condition is there, there we need some salt tolerant varieties. Okay, and where there is a rain-fed condition, and we need some varieties which can tolerate such submergence where they can tolerate the high amount of rain. The major proportion of the cropped area in the country is the rain-fed. Okay, and if we go for estimation that the soil affected areas of India is about 7 to 20 million hectare, and which about 2 point and about 2.8 million hectares are the alkaline soil. So for those we need if we need to utilize those areas, in the previous video we have talked that we can't increase our net cropped area, but this way we can utilize our uh, not used area as by developing new varieties for which are tolerant to those conditions. Most of these areas are spread in the states of UP, Haryana, and Punjab. After that, we can eliminate the toxic substances. It is one of the major objectives in our breeding program. These toxic substances often reduce the value of the crop, and if we can eliminate them, it will be the safe for our consumption. Like in case of the khesari, the one important thing that is the boa. Often it is one of the neurotoxin which cause the paralysis. Similarly, in case of brassica, there is a erucic acid. We have developed some varieties which are uh, zero double zero variety, which are now in commercially used. Removal of such toxic substance will increase the nutritional value of these crops, obviously. After that, we will see some achievements, what we have achieved as of now through this plant breeding one by one. In case of wheat, we have seen the semi-dwarf wheat. The semi-dwarf wheat was developed by Normani Borlaug, you all know, and he developed it in the CIMET, Mexico, that is the International Center for Wheat and Maize Improvement, that is CIMET. After that, he used the Japanese variety Norin 10. It is often asked in the exam that which variety is used as a source of dwarfing gene. There is the Norin 10. This is one. This one is the variety, not the gene. Okay. After that, if we see that in 1963, ICR introduced several semi-dwarf selections from the CIMET. From CIMET to the India, ICR have introduced some varieties like Kullan Sona and Sonalika. These were about the primary introduction. After that, the semi-dwarf wheat varieties, what are the qualities they possess? That they are lodging resistance, fertilizer responsive, and high yielding. That makes them different from other varieties. They are generally resistant to rust and major diseases of wheat, and wheat due to the incorporation of the resistance gene into their genotype. In addition, they are also photoinsensitive, and many of them are suitable for late planting. That means these varieties are enabling the cultivation of wheat in non-traditional areas like West Bengal, which we have talked about previously. These wheat varieties have results greatly increased and stabilized wheat production in our country. If we see the white dwarfing gene, that is the reduced height 1, reduced height 2, or RH1 plus RH2 leads to increase in about at least 15% overall increase than over the tall isogenic lines. That means the lines, what we mean by the isogenic lines, that means if we see this is the dwarf variety and if it is the tall variety, they will differentiate only because of this RH1 gene or RH2 gene. It will possess the RH1 gene, except all are the same, only this will possess the RH1 RH1 gene or RH2 gene, and this will lack them, and it will have the greater than 50% of yield increase because of these genes. Semi-dwarf wheat varieties are also marked increase in the harvest index. If we ask in the true false, you must say this is true that semi-dwarf wheat varieties are also seems to be increasing the harvest index as compared to the tall varieties. but this is regarded as a side effect of the RST gene rather than their main effect. Their main effect is the semi-dwarfing. Okay. In contrast to RST2, RST1 and RST1 and RST2, RST3 and RST18, RST3 and RST18, they do not significantly increase the yield, but they provide the good degree of lodging resistance. So it is a very good point to remember that this RH2, RHT1 and RHT2, these two genes are mainly go for increasing the yield, and they are mainly responsible for this dwarfing. But whereas in RHT3 and RHT18, RHT8, sorry, RHT8, they are mainly responsible for the lodging resistance, but not yield. Okay, this you should remember. After that, let me clear. After that, if we see there's a for this RST gene, they are the mainly the DELA domain proteins. Okay, so they actually replace the transcription of gibberellin responsive genes. You all know that the gibberellin is a hormone which is responsible for the internode growth. So if we suppress, that means there is a two node. This growth is inhibited by this DELA domain, this RST genes. Okay, sorry. This RST genes encode the DELA domain protein, which actually replace the transcription of gibberellin responsive genes, and that is how they are dwarfing the wheat plant. In and we also seen that the semi-dwarf wheat varieties are also photoperiod insensitive, and this photoperiod insensitivity is due to the presence of genes that is the PPD1 and the PPD2. And if we see the best combination, we will find that if there is a in variety that is present is RHT1 plus PPD1 or PPD2 is present, or if we see that is the RHT2 plus PPD1 or PPD2 is present. So these two will be the best combination and for semi-dwarf wheat varieties. Whereas if we see both, if there is present PPD1 and PPD2 both present together in such line, there is a generally lower the yield of than the others. Okay, so there must be present only one, that is if there is a PPD1 present, and if there is present also PPD2, that means if though we both are present, they will lower the yield. If they present alone, they will increase in contribute in the yield also. Then, if we talk about the semi-dwarf rice varieties, this variety was developed from the variety that is the DGU gene, that is the DGWG, which is the dwarf and early maturing variety of Japonica rice, and which was from Taiwan. Taichung native one was developed in Taiwan and using this DG Ujin variety, and IR8, which was developed in IRRI International Rice Research Institute Philippines, which were introduced in India in 1966. They are extensively grown in few years, but they are later replaced by superior semi-dwarf varieties like Joya, Ratna, etc. If we see, this is the miracle rice. We call it as a miracle rice, and Indian miracle rice we call as a Joya. The semi-dwarf rice variety were the lodging resistance, fertilizer responsive, high yielding, and photoinsensitive. This photoinsensitivity has allowed the rice cultivation in the non-traditional area like Punjab, even in traditional areas, light wheat rotation also become possible due to this. We have developed this Ratna variety due to a TKM6 and IR8 crossing. Then the most important one in this lecture is the nobilization of Indian canes. It was done by Venkataraman, whom we called the sugarcane wizard. Okay, um, the Indian canes, the scientific name you should remember, there is the Saccharum barbari, which is the Indian or which is the largely grown in case of North India, and they are hardy but poor yield in sugar content. They are poor yielder, and sugar content was also less. But the tropical noble canes, that is the Saccharum officinarum, which is having the thicker stem, higher sugar content. But they performed very bad in case of North India, primarily due to the low winter temperature in this region. So C.A. Barber and T.S. Venkataraman, what they did in this SBI, there is a sugarcane breeding institute, that is present in the Coimbatore. They transferred this trait, that is the thicker stem and higher sugar content and other desirable characters from the noble canes to the Indian canes. That we called as a nobilization of sugarcane. That means they caused the noble cane to the Indian cane, that is the Saccharum officinarum into Saccharum barberry, and they transferred this desirable character like thicker stem and high sugar content into the Indian canes, and that is what we call as the nobilization of Indian canes. They also caused the Saccharum spontaneum, a wild species, to transfer the disease resistance. This may come in the matching that what they have transferred. The disease resistance from the Saccharum spontaneum and higher thicker stem and higher sugar content from the Saccharum officinarum. Okay. After that, in case of hybrid millet, we can see uh, that there was the hybrid maize development which have occurred in the 1957 in the collaboration of Rockefeller and Ford Foundation. As a result of hybrid varieties were developed in 1961. That means you all know that AICIP. AICIP was launched. AICIP maize was launched in 1957, and 1961 there was a development of the hybrid varieties. Subsequently, several hybrids, mainly double cross hybrids, are developed. Hybrid maize become very popular in some states like Karnataka and other states also. But therefore the composite varieties like Manjari, Bikram, Sona, Kisan, CO1, and NLD, Renuka also being developed in this session. After that, if we see that these are all talking about the maize that developed the hybrid. There is a single cross hybrid. That means it was the first they have developed the double cross hybrid. After that, the composite variety. And now, if we talk about the single cross hybrid, we have got the Bibek Maize Hybrid 9, Malvia Hybrid Makka 2, Bibek QPM 9, etc. Okay, the hybrid maize about yielded about 40% more than the local open varieties. That is the CSH1. Uh, Development of hybrid sorghum and pearl millet also began in the 1961, and the first hybrid varieties of CSH1 was developed in sorghum. Sorry, this was wrong. The CSH1 was this in case of sorghum it was developed, and in case of bajra it was about HB1, hybrid bajra 1, released in 1964. And since then several hybrid varieties also developed in case of jowar like CSH2, 3, and so on, and in Bajra PHB 10, PHB 14, etc. After that, if we see that the hybrid cotton on India has achieved the commercially exploiting heterosis in cotton. In India, cotton is one of the most extensively commercial crop which exploits the heterosis breeding. The first hybrid variety of cotton, there is a H4, which was hybrid of the two Gossypium hirsutum strain. It was developed in 1970 in the GAU, that is the Gujarat Agricultural University, Surat Station. Other hybrid varieties like JK JK hybrid 1, Godavari, Sugana H6, etc., which was all about Gossypium hirsutum hybrid varieties. That means the intervarietal cross. Bharalakshmi, CB156, Savitri Jayalakshmi, H2HC. These all varieties were developed by interspecies cross. That is means the Gossypium hirsutum into Gossypium barbadense. The hybrid varieties are high yielding and have high gaining outturn, good fiber qualities. That means they are possessing all the good qualities than the open pollinated one. So we have they earlier we have developed also the hand pollinated varieties like G-cot DH1, G-cot DH9 also. These are two hybrid varieties of desi cotton. Okay. If we talk about the molecular breeding, the marker assisted breeding enables the dependable indirect selection of the desirable genes using highly reproductive DNA markers. That means we use the DNA marker technology to isolate our or to select our desirable plant, and this we called as a marker assisted selection. We have some examples for this success. There is a bacterial leaf blight in case of rice varieties, we have developed the improved Pusa Basmati 1 and improved Samba Masuri. Which are bacterial leaf blight BLB resistance. Whereas in case of submerged tolerance, we have Sorno Sab 1. We have transferred this Sab 1 gene to the Sorna variety. Improved protein quality measures as a Vivek QPM1, QPN9. We have developed using this marker assisted selection. In addition, the genome sequencing is also one of the component of the molecular breeding, which we have gained so much success. There are sequences of Arabidopsis thaliana, which was done in 2000. In case of rice, the first draft was produced in or published in 2002. In case of PGNP, it was done in 2011. Intensive efforts have been made to use this genomics and genetic transformation for identification and confirmation of the allele contributing to the desired traits and to develop the molecular markers for their marker-added selection. This approach is expected to enhance the efficiency of the breeding efforts by improving difficult to select for complex traits. Uh, that means this approach will help us in selecting those complex traits which are generally impossible to select by naked eye. We need some evidence, and these molecular approaches are giving such uh, evidence. And marker assisted recurrent selection has coupled with the off-season crops has permitted 2 to 3 cycles selection cycles in 1 year. That means we can select our desired crop or desired line or desired plant by seeing their DNA. Okay, so we no need to grow them for a full season or etc. So we can go for 2 to 3 selection cycle in 1 year. Okay, whereas in conventional approach, we need one selection cycle in 2 to 3 years, which is very difficult. The scheme called genomic selection is designed to use marker distributed over the entire genome, which to effectively select the specific quantitative traits. That means if we have to select the quantitative traits, it is very much difficult. And this genomic selection also helping us to select these quantitative traits using the markers which are distributed all over the entire genome. Next, if we see about the transgenic varieties, that means the recombinant DNA technology, which have been played the most important role in case of plant breeding, uh, where it is the most powerful tool for creating the crops with novel desired phenotypes, or as we call it as a designer crop. That means we can design our plant breeding according to our needs, and we can incorporate our desired genotype or gene from an unrelated organisms into our crop varieties, which can help us in providing the better resistance, say better yield, etc. Such as in case of the BT cotton, on which they are more resistant to the lepidopteran insects, and that makes them more insect resistant and more yield. In 2007, about 131 different BT cotton varieties were released. In 2009, the total area under the crop such hybrids was 8.4 million hectares. Recently, an indigenous insect varieties, that is the Bikaneri Narma, and a hybrid NHH4, which also have been also released. It is noteworthy that the India has now become a cotton exporter from being a net importer. In 2003 and 4, they import cotton from outside, whereas now, if we can say that they are more than self-reliant, that is, they are the exporter to the other countries. Because this is possible due to the cultivation of this BT cotton. And for this, the BT cotton is highest cultivated in India too. There are other varieties also like BT Binjal is also under commercial cultivation. If we see in India, these are all, not all, but many of the cases, if we see, they're developed in the outside India. But if we see that some of the cases in India, that is the improved varieties, generally about 15 to 80% of the existing all local varieties were developed, and these often yields much higher than the local varieties. Due to the resistance to disease, the improved varieties save the cost of the plant protection, stabilize the crop yields, and in addition, they offer the numerous other advantages too, which we have discussed so far our video. And these all are the Indian achievements. And if we see the overall, there's a 50 to 80% overall increase in the existing local variety yield, and there are many disease resistance, etc. Thus, the plant breeding is immense economic importance to a nation like India, which is an underdeveloping country and which is actually which has the agriculture-based economy. In view of this, the ICR New Delhi has launched elaborate and well-developed uh, AICRP, that is the All India Coordinated Research Project for almost all important crops, which are actually involved in improving such particular crop day by day. Till now, we have seen some good signs. Okay, some good things about plant breeding. That means which in which they are contributing, but there is a bad side always. That means there are some undesirable consequences of plant breeding too, which we have to face in our upcoming days. That means crop improvement, which lead to development of many desirable changes in the species, which make these crops useful to the humans. But it also generates some undesirable consequences, which are those: firstly, the genetic erosion, narrow genetic base, increase the susceptibility to the previously minor disease, which are previously we see them as a minor disease, but now they are becoming more major diseases and causing harm to the crop. Then the last one is the yield plateau. If we see one by one, the first one is the genetic erosion, where if we see that the reduction in the variability among their cultivated varieties, that means the variability में रिडक्शन आ जाती है. ठीक है, तो वेरिएबिलिटी अगर घट जाती है तो हम हमारा जो सिलेक्शन का जो सोर्स था वो कम हो जाएगा. ठीक है, दिस रिडक्शन इज प्रोड्यूस्ड बाय टू फॉलोइंग factors. First one is the replacement of the numerous indigenous local varieties by few more homogeneous and improved varieties. Use of similar or related varieties as parents in breeding program. That means what we can see that the local or improved local varieties which were mainly contributing uh, as a source of genetic variation, we are replacing those varieties with our more improved more improved homogeneous improved lines, like in case of self-pollinated, which are the pure lines. We know that is the pure lines are the progeny of the single plant of the self-pollinated cross species. Whereas in the hybrid, this is also the single genotype. Okay, so what we are doing, we are diminishing the, we are undesirabling the, we are not selecting the variable population. We are going for the improved and homogeneous population. And by this, what we are doing, we are diminishing the variability. Okay, and in the second point we have talked that we are using such say one parent which is good, one good parent in developing in many varieties. So if the parent is the same, if in some cases if the one disease is susceptible becomes susceptible to that parent, then means the all our variety will be susceptible to the diseases. So this will uh, affect our breeding program. A small number of improved varieties in each crop become predominant and rapidly replace the heterogeneous local varieties, leading to a rapid depletion of the genetic variability, which we call as a genetic erosion. This in turn limits the prospects of further improvement in the crop varieties, because if the variability is the main prerequisite of any modification of the characteristics. अगर वेरिएबिलिटी प्रेजेंट ही नहीं रहेगा तो अगर किसी क्रॉप में डिजीज आ गया, नया कोई डिजीज आ गया, तो हमारी क्रॉप बस वो एक ही सैंपल है उस वैरायटी का और कोई डिजीज रेजिस्टेंट सैंपल ही नहीं है, तो हम कैसे उसको इस्तेमाल करेंगे? अगर मेरे पास डिजीज रेजिस्टेंट सैंपल रहेगा, दैट मीन्स वेरिएबिलिटी हमारे पास प्रेजेंट रहेगा, तभी तो मैं इसको इस्तेमाल कर सकता हूँ ना। तो अगर नहीं प्रेजेंट रहा तो हम उसको कैसे इस्तेमाल कर सकते हैं? तो इसी वजह से दिस वी कॉल्ड एज अ जेनेटिक इरोशन. After that, how we can uh, improve our genetic stocks, like we can go for germplasm collection, which can minimize this detrimental effect of the genetic erosion by collecting and preserving the variability which are present in the crop in their related species and wild species. What we can do, we can collect those variable plant species and we can conserve them. Okay. The next one is the narrow genetic base. That means what we mean by the genetic base. Genetic base is the genetic variability present among the cultivated varieties of the crop species. That means कितना genetic variability present है उसको ही हम बोलते हैं genetic base. Many of the improved varieties of a given crop have one or more parents. That means हम देख सकते हैं कि बहुत ऐसे variety होगी जिसका parent same रहेगा. Okay, so for example, in case of rice, we can see that the uh, iodate TN1 as their we can see there's the 17s and 80s. In case of 70s and 80s, there's the iodate and TN1 is very much predominant and promising varieties in those days. And these iodate and TN1, which themselves are related to each other by common parent like DGU gene as a source of semi-dwarfing gene. So if we can see, if this variety gets susceptible, this will also be susceptible and this will also be susceptible. Okay, so this is how we are the narrow genetic base is contributing in our plant breeding. Similarly, almost all semi-dwarf wheat varieties के लिए भी हम अगर देखें तो RST1 gene, RST2 gene, these all genes comes from the Norrin-10 variety and it was predominantly used in the breeding program. Okay, thus improved varieties of a cross species becoming increasingly similar to each other due to the commonness of one or more parents in their ancestry, and this has led the narrowing down the genetic base of these crop varieties. Now what we can do? The narrow genetic base has created genetic variability which refers to the susceptibility of the most of the cultivated varieties of a cross species to diseases, insect pests and other stress due to the similarity in the genotype, which we have seen in this slide. But what we can do, we can search for new dwarf gene or new variability, which and make use those new genes or new source of genes in our crop variety, and we can incorporate for them for further use. Now, if we see an example of genetic vulnerability, if we see there is an outbreak of epidemic uh, in case of southern leaf blight, that is Helminthosporium maedis in case of maize in USA in 1970s, where the all we used that the due to the Texas male sterility CMST hybrid, which was become susceptible to the T rays of the leaf blight pathogen of the Helminthosporium maedis. And if we say in brief that in 1970s most of the maize varieties was developed, hybrid varieties was developed using this CMS T cytoplasm, okay, Texas male sterile cytoplasm as a female parent, but this cytoplasm was very became susceptible to the T rays of the Helminthosporium maedis, and this became very much susceptible, and this destroys the maize in USA due to the southern leaf blight. Okay, it about 75 to 80% of the maize was occupied by this hybrid, and you can understand by this that the 75 to 80% of the maize was destroyed. Similar case was reported in India too. There is a susceptibility to the Bajra hybrid due to the Bajra hybrids due to the down mildew and ergot. Again, this susceptibility was contributed by the male sterile parent of these hybrids, that is the TIP23A, and its dwarf derivatives, that is the TIP23 dwarf A. Okay, these varieties were susceptible to these diseases, and this makes the huge epidemic in India. But in this case, the susceptibility was due to the nuclear genes, not due to the cytoplasmic genes. This genetic vulnerability can be avoided by using the diverse and unrelated parents in the breeding program, or by using the unrelated source of male sterility and semi-dwarfness. Okay, if we use different sorts of semi-dwarfness and male sterility, etc., in case of our hybrid breeding program, we can avoid this loss of genetic vulnerability. Then there may be increased susceptibility in the minor diseases. That means those diseases which are minor in previously, but now they are very much dominating. We can see that in case of uh, botrytis cinerea, botrytis cinerea, we see in case of the botrytis cinerea, there is the gray mold in case of chickpea was developed in 1980 to 1981, and 1981 to 1982. Oh, in case of Punjab, Haryana, and parts of UP and Bihar, this was previously supposed to be as a minor disease, and we have not much emphasized on that. And there are many other diseases which are mostly prevalent, which have we have developed resistant varieties to them, and in the meantime, this pathogen become very much dominant and become major diseases in these areas. Another example of such thing, there is the cardinal plant, which was there is the Tiletia indica in some case, some wheat varieties like WL711, which became very much active. After that, if we see the yield plateau, that means uh, if we make a graph, we can see it very clearly. That is a with time. Suppose this is a graph, it is a time and it is the yield. With time, that is a this. Suppose this is a variety A, and its yield become increase, but with time, its popularity, it will get a plateau. That means it yield will be stabilized. Okay, this we call this as a yield plateau. Okay, sooner or later the variability of the yield in cross species is exhausted. No, that means no further yield increase is obtained. That means yield reaches into a plateau. That means no further yield is increased. That uh, suppose here at first the variety yield up to suppose 20 kg, 120 kg kilo. Okay. After some time, if we do some modification, and this will is up to this. After that, in some time, it will giving about 200 kg yield. Okay, after that, if we are doing something, but it will not producing any extra yield, so this situation we call termed as an yield plateau. Such parents are evident in case such plateaus were evident in case of wheat and rice, which was after the exploration, before the exploration of the semi-drop genes and being experienced once again. That means before we have incorporated the semi-dwarf gene, we have seen such plateaus, that is yield are not increasing. Then after we have incorporated, uh, we after we have incorporated, that means in this case what happened? The graph will be like this. That means at first, before 1960s, the yield was getting deeply static. But after the increase, but after when we have incorporated the semi-dwarf gene, it have increases a lot. And now in 2020 and 30, it have again become the plateau. It is about 1960s time, and it is about 2023 and so on. Okay. So in such cases, the new variety has to be introduced in breeding population in order to break this plateau. That means we have to find out new variability. Okay, there is an increase in the evidence that the wild relatives of the cultivated species may provide yield genes. That means we have to, if we need something, uh, we have we don't know. Suppose we don't know anything, we go to our elders like Dada Nani to know them, to ask them what is this. So same is the case in case of crop varieties. We have lacking the yield genes, we have to pray for the yield genes to the wild relatives of the cultivated species. If they have, we can get the yield genes for the breaking this yield plateau, like this one. If we get some yield genes, then yield genes, if we get some, then we will experience and again an increase in the we will experience again an huge amount of increase in the crop yield. In some cases, the novel breeding approaches must be useful, like development of hybrid varieties in case of rice, which we have discussed, which is actually is the method by which the breeding procedures is progressing. Okay, so this was all about, and thank you for your presence. See you in the next video.